Welcome to This Explainer. I am so thrilled you're here today because we are going to dive straight into one of the absolute wildest challenges in modern science. We are looking at how we can actually peer inside the human mind in real time without ever opening the skull. We're going to unpack the incredible cutting-edge technologies that allow us to decode brain activity entirely non-invasively. Just think about this for a second. How do we actually capture profound, super fleeting psychological phenomenon on a scientific level? I mean, whether a person is trying to recall a deeply hidden childhood memory, formulating a deceptive lie, or literally approaching the very brink of death, the brain is firing off incredibly complex sequences of signals. Catching these signals as they happen seems almost impossible, right? Yet, cognitive neuroscience has somehow found a way. To really wrap our heads around the massive stakes of what we're trying to measure here, look at this astonishing number. A Gallup poll actually found that 15% of people stated they've had a near-death experience or an NDE-type event. We're not just talking about studying simple reflexes. We are trying to decode profoundly deep, complex, and sometimes totally life-altering human experiences. The actual phenomenology of a near-death experience really shows us exactly what scientists are up against. People report seeing phosphenes, those intense flashes of light, having super vivid out-of-body experiences, traveling down a dark tunnel, or suddenly experiencing a panoramic life review. These are immensely complex, deeply vivid cognitive events. They involve memory, vision, emotion, and spatial awareness, all happening in a brain that might be under extreme physical stress. So, how on earth do you map that? Okay, let's dive into this. To capture these profound, fleeting moments in the living brain, researchers essentially need giant, highly advanced, non-invasive cameras. We need tools that can literally look right through the bone and tissue to see the biological sparks of human consciousness, completely without altering the brain itself. But as you'll see, designing the perfect brain camera means solving a massive puzzle in engineering and physics. Part 1. The two big questions: the map and the stopwatch. The whole puzzle of neuroimaging essentially boils down to two overarching questions that scientists are constantly fighting to answer. Where is the brain activity happening, and when is it happening? These are two totally distinct parameters. On the spatial side, we have our map. This dictates how accurately we can pinpoint the exact millimeter of brain tissue that is firing. On the temporal side, we have our stopwatch. This dictates whether we can catch an electrical signal in the exact millisecond it happens, or if our measurement ends up blurry and delayed. And that brings us to a fundamental reality in cognitive neuroscience: the trade-off. There is simply no perfect all-seeing machine. Because of the strict laws of physics and biology, a neuroimaging technique usually excels at either mapping the precise location or capturing the exact timing, but almost never both at the same time. It's a strategic game of choosing the absolute right tool for the specific question you're asking. Part 2. Stopwatches, EEG and MEG. All right, let's look at the tools that prioritize time. These technologies are entirely focused on catching the incredibly rapid, millisecond by millisecond electrical chatter of the brain. The most famous of these is EEG. It works by placing electrodes on the scalp to measure tiny changes in electrical activity. But, as you can imagine, background brain noise can make this super messy. The brilliant solution is something called event-related potentials, or ERPs. By repeating a stimulus, like a sound or an image, dozens of times and averaging the recordings together, researchers can basically wash out the background noise. This isolates a single waveform that shows exactly what the brain is doing, with a temporal resolution of just 1 single millisecond. But remember the trade-off. Let's compare EEG to its magnetic cousin MEG, which measures the magnetic fields produced by that exact same electrical activity. Both are incredible stopwatches, but they really struggle with the map. EEG picks up electrical volume currents from all over the brain, but the thick skull severely distorts those fields, giving us a spatial resolution of a very blurry 2 centimeters. MEG has clearer signals because the skull is totally transparent to magnetism, but it's actually blind to currents on the ridges of the brain, called gyri. For both of these, solving the inverse problem, basically working backward from the outside of the skull to find the exact source inside, is incredibly difficult. Actually, scratch that. It's not just about the physics, it's about the humans inside the machine. Despite the incredible temporal precision of MEG, there's a surprising catch. Studies have found that 35% of people actually find the expensive, restrictive MEG machine a bit upsetting, or they even experience mild side effects like muscle aches and headaches. So, participant comfort is a very real factor in pulling off this research. Part 3.
maps, PET, and fMRI.
So the crucial point is, if we desperately need to know the exact millimeter of activity, we have to put away our stopwatches and pull out our maps. These map-making tools are what we call indirect measures. They don't measure the actual electrical sparks of neurons firing at all. Instead, they track the brain's plumbing. When a brain area works hard, it needs more oxygen and glucose, so blood rushes to that area. These tools measure that sluggish blood flow. Because blood takes seconds to arrive, the temporal resolution, the stopwatch part, is frankly terrible. But the spatial map, it's beautiful.
An older version of this is the PET scan. It requires injecting a tiny amount of a radioactive isotope, like radioactively labeled water, straight into the bloodstream. As the isotope decays, the machine detects it. But get this, the delay between the actual brain activity and the PET signal peak can be up to a full minute. Because of this awful temporal resolution and obviously the invasiveness of radiation, PET is now mostly used for vital clinical tracking, like mapping nicotine receptors or disease progression, rather than real-time cognitive research. And this brilliantly illustrates the modern gold standard for mapping, fMRI. This involves a massive scanner housing a magnet that can weigh up to 11 tons. It tracks the blood oxygen level dependent, or BOLD signal. Now, while its stopwatch is definitely poor, lagging about 2 to 6 seconds behind the actual thought, its spatial map is nothing short of stunning. It allows us to pinpoint activity in a volume of tissue as small as 1 single cubic millimeter.
Part 4. The resolution spectrum.
Let's move to and see how this all builds together. If we compare these tools side by side, we see a really clear divide. EEG and MEG measure electrical and magnetic signals directly. Their stopwatch is excellent, at exactly 1 millisecond, but their map ranges from fair to poor. Conversely, fMRI and PET measure indirect blood flow. Their stopwatch is poor to very poor, lagging by seconds or even a full minute. But the fMRI gives us an excellent, highly detailed map. Part 5. Choosing the right tool. So, what does this incredibly diverse toolkit actually mean for us? It means that before a single scan is ever run, researchers must deeply analyze the specific cognitive phenomenon they want to study. Your key takeaways here are first, you gotta embrace that no perfect machine exists. If you are studying how quickly the brain detects a familiar face, where timing and the exact order of operations are absolutely everything, you must use EEG or MEG. But if you want to identify the exact subnetwork of brain regions involved in complex problem solving, you need the spatial mapping of fMRI. The scientific question always dictates the technology. We can only understand the mind by cleverly matching our questions to the limits of our cameras. Which leaves us with one final, super exciting mystery to ponder. As engineering and physics continue to advance at a breathtaking pace, what future technologies will finally break this trade-off? What wild invention will eventually allow us to map the precise millimeter of the brain at the exact millisecond of a thought, perfectly capturing the deepest secrets of human consciousness? Thank you so much for joining me on this explainer. I hope it sparked your curiosity, and I highly encourage you to keep exploring the incredible world of cognitive neuroscience.